Good morning, students. Today we are start with our topic with the structure of human eye, and later that we will learn about the functioning of human eye. This is the picture of picture showing the detailed structure of eye, and the eye is composed of many layers. So we are going to see which are these layers. The outermost layer is called sclera, or otherwise sclerotic cord. This is the name of the outermost layer of the eye. Sclera, it's given here, sclera. And this sclera, what's the peculiarity, is made of dense fibrous connective tissues. What's that? Sclera is made of dense fibrous connective tissues, and also the sclera is very opaque. Opaque means it will not permit the passage of any light. But when it comes to the front side, the sclera became converted into a transparent layer called cornea. It will permit the entrance of light to this portion, cornea. And towards the front side of the cornea, there present another transparent layer, protective layer for eye. Its name is conjunctiva. What's the name? Conjunctiva, spelling CONJUNCTIVA. So, up to here, what have we learned? The first layer of the eye is called sclera, and is composed of dense fibrous connective tissue, and is a very opaque layer. But towards the front side, the sclera become transparent, and the transparent portion of sclera is called cornea, that permits light to enter into the eyes. And the covering to the cornea in the front side present another transparent layer called conjunctiva. Okay, clear. Then we are going to the next layer. Next, this red colored layer. Its name is choroid. What's the name? Its name is choroid. What's the peculiarity of choroid? It is a pigmented layer, right? It's a pigmented layer and is highly vascular. Vascular means it is richly supplied with the blood capillaries. Blood capillary rich layer ആണ് this choroid. And also since it is highly vascular, it is the layer responsible to give nourishment to the eye, because much blood capillaries are present in this layer. It is providing nourishment. We know, we are already learned in other chapters like nutritional physiology. We have learned that through blood we are getting almost all nutritions. Almost all nutritions are providing to each and every cell by the blood capillary. So as this layer is rich in blood capillaries, it can give nourishment to the entire eye. And also it is responsible for some amount of light capture, light absorption also. That's about choroid. And one more peculiar thing regarding choroid is that towards the front side, the choroid separate from sclera and to form a peculiar diaphragmatic structure, a partition, a separate membranous structure separated from sclera. The name of that extended membranous diaphragm-like structure is called iris. Iris means in this picture is very clear. This light blue color is the iris. Iris is actually a part of choroid layer. Towards the front side, choroid separate from the outer sclera layer and form a diaphragm. That diaphragm-like part of this choroid is called iris. And this iris contains so many muscles. Iris contains so many muscles are present in the iris. And with the help of these muscles, and also one more point here, and the center of the iris there's present a small opening, small space called pupil. Here we can see that. And a small opening is there in the center of iris, is called pupil. That's about iris. Iris is the diaphragm-like portion that present in front of the towards the front side of the eye, the part of choroid. It is at that iris part the choroid separate from sclera and form a thin diaphragm-like structure called iris. This and next is at the junction of iris with the choroid, it becomes somewhat bulged. It is somewhat bulged to form another structure called, it's a thickened portion. Here we can see it's a thickened portion called a ciliary body, and it's with so many muscles are present inside this ciliary body. Ciliary muscles, because of the contraction of ciliary muscles, we can alter the shape of the lens that's present inside the eyes. By the contraction and relaxation of muscles present in the ciliary muscles, we can regulate the shape of the lens. And by regulating the shape of the lens, making it elongated and making it shortened, according to the need of accommodation, whether we are viewing a far object, if we are viewing a nearest object, according to that the lens shape need to be adjusted. That adjustment is done by the contraction and the relaxation of muscles present in the ciliary body. And one more thing I missed here to tell you, that's about the location of the eyes pupil. Eyes, actually it is located in the socket present in the skull, right? And in that skull, in that socket, it is its movement, eyeball's movement is regulated by 6 set of extrinsic muscles. And the eyeball is located in a socket the present in the skull, and the eyeball's movement is controlled by 6 set of extrinsic muscles that I actually missed to tell you in the beginning. Okay, that's about the choroid and sclera. What are the points we need to remember? Sclera is opaque. Towards the front side, it becomes transparent called cornea. And cornea is again covered by another protective transparent layer called conjunctiva. Then coming to choroid, has peculiarities pigmented and vascular. Since it is vascular, it gives nourishment to the cell, nourishment to the eyes, and also it helps in the absorption of light. Towards the front side, the choroid separate from sclera and form a diaphragm called the iris. And the iris consists of so many muscles, and with the constriction of this iris muscles helps the constriction and dilation of pupil. That we can very clearly see if a torch light is pointing towards our eyes, we can see that our pupil will constrict to prevent only a few light to enter into the eyes. That's a function of iris. Iris constrict, by the constriction of iris muscles, it can constrict or enlarge the size of the pupil. And at the junction of iris, there present an expanded portion, a thickened portion called ciliary body, and the ciliary body consists of many muscles. Ciliary muscles are present. By the contraction and relaxation of ciliary muscles, we can regulate the shape of the lens to achieve power of accommodation. This is about the structure. Then exists one more layer. It's a very important layer that helps in vision. That's innermost layer. It's called retina. So totally there is 3 layers. Innermost layer is called retina, and retina itself consists of 2 layers. An outer pigment layer and an inner nervous layer is there. So that we can see in the next slide. This is the retina. This is a red portion, retina, and its enlarged view is given here. An outermost layer is there and an innermost layer is there. Outermost layer is the pigment layer, and the innermost layer, innermost layer is the nervous layer. And this nervous layer contains almost all rods, and cones. Rods are the cells responsible to vision in dim light. Cones are the cells responsible for vision in bright light. Right? And regarding the arrangements of rods and cones in this inner layer, we need to learn something. What about that? The rod, whatever it be, rod cell or cone cell, its location is somewhat here, and it is synapsing with a bipolar neuron. Bipolar neuron means what we already learned in the neural physiology. Bipolar neurons are neurons having two extensions. It is synapsing rods or cones. It's synapsing with a bipolar cell, and this bipolar cell in turn is synapsing with what, glial cell. Glial cell. Here is very easy to understand. This is the rod or a cone cell. It is synapsing with the bipolar cell. Bipolar cell in turn is synapsing with a glial cell, and from the glial cell emerge the nerve. The branch of nerve is emanating from the glial cell, and all these nerves unite to form a single optic nerve. All these nerve endings from each glial cell unite to form a single optic nerve and collect the information to the brain, and we interpret the image that we are observing. This is actually happening. So, in addition to all these type of neurons, some other neurons also present. Its name are amacrine cell and horizontal cell. What's it? Amacrine cell and horizontal cell. Spelling amacrine means, let me check whether this picture is given here or here. Here we can very clearly see that. Once more I will explain to you. Here is the this is the cone cell and this is the rod cell. Anyway, both of them synapse with the bipolar neuron. Bipolar neuron in turn is synapsing with the glial cell, and from its base neurons are emerging, and all these neurons emerging from all glial cells unite to form a single optic nerve. And in addition to this present, which are the additional neuronal cells, they are called horizontal cells and amacrine cells. Where is location? Amacrine cells and horizontal cell. Horizontal cells actually present in the region where the cone cell synapse with bipolar cells. A horizontal cell. Where horizontal cell is located? It's located at the synapsis point between rod or cone cell with the bipolar neurons. And when where this amacrine cell is present, amacrine cell is present at the junction where the bipolar neuron synapse with the glial cell. All these interconnections are letting the eyes to convey the signal of vision very efficiently to the brain. Then next, another 2 point we need to understand: foveae and blind spot. Foveae, what is foveae? Foveae is that region in retina. At that region, cones number is very high. Cones rich retina's geometric center is called foveae. At that point, the ability to vision, bright light vision is very high because cones are more at that point. So what is foveae? Foveae is cone rich retina's geometric center. This is this point. It's a notch like a small depression, and this portion cones number is very high. So that region is called foveae. Then what is blind spot? Blind spot means the region where all the neurons converge to form an optic nerve. The point where that optic nerve is formed, at that portion rods and cones are totally absent. At that point is called a blind spot. Understand? Blind spot are blind spot are those points in the retina where all the nerve fibers converge to form an optic nerve. At that converging point, rods and cones are totally absent. That point is called a blind spot. Okay, next, and the image that is formed in the retina is of inverted. So that retina is called the inverted retina, and that inverted means what? Well, if we are visualizing something, that image that formed in the retina is inverted. Means Inverted, I think you understand. Inverted image is formed in the retina, and that inverted image is interpreted, and we are understanding that image in the correct form according to the function of that brain. Brain actually interpret and correct that image to again to that real image. So, here we can see that retina is inverted retina. The image of this girl is actually formed in this inverted form in this retina, and this inverted image is corrected after reaching the brain. Then two more terms, aqueous chamber and vitreous chamber. Aqueous chamber, which is the A means it's a beginning, first letter. So first chamber means space between the cornea and the lens. That chamber, first chamber. Because first is aqueous chamber. A is for first letter. So it belongs to first chamber. In that way we can memorize that. Aqueous chamber is the first chamber. Chamber means it is present here between lens and cornea, and beyond the lens there present another big chamber called the vitreous chamber, and aqueous chamber is filled with aqueous humor, and vitreous chamber is filled with vitreous humor. Spelling I will give you. Spelling is given here. Aqueous chamber and vitreous chamber. And humor, HUMOUR. Vitreous humor is a fluid filled in the vitreous chamber, and aqueous humor is the fluid filled in the vitreous chamber. Humor spelling HUMOUR. Okay. Next, we are going to understand about the ultrastructure of rods and cones. This is rods, and this is the cone cell. Both of them have an outer segment, outer segment and an inner segment. Then what's the peculiarity of these outer segments and inner segments? And the outer segment is the light sensitive segment. It's a peculiarity. It is the light sensitive segment, and inside this outer light sensitive segment present so many visual pigments called rhodopsin. Rhodopsin spelling RHODOPSIN. Rhodopsin cell. Rhodopsin you have learned in some other chapter. Anybody is remembering? Rhodopsin. Where? Vitamin A deficiency diseases. We have learned about rhodopsin. Where? Okay, rhodopsin. It's a protein composed of scotopsin and retinol. I think you remember that. Okay, leave it. Anyway, the outer light sensitive region contains so many lamellae. In that lamellae present the visual pigment rhodopsin. And coming towards the inner segment. Inner segment contains nucleus and some other cellular organelles, and it is synapsing with the bipolar neurons that we already learned. This is about the ultrastructure of rods and cones. Outer segment is there, inner segment is synaptic ending. It synapses with the bipolar neurons, and inside the inner segment so many photo visual pigment called rhodopsin is present. It is arranged in lamellas inside the outer light sensitive segment. Okay. Next, difference between rods and cones is oftenly asked for your exam. So, need to learn very well. Don't avoid it. Rods and cones. It's larger. In the case of rods, outer segment is larger. Case of cones, outer segment is smaller. That you can understand on going back and see the image of rods and cones. And regarding the number, rods number is 100 million, and the number of cones is 3 million, less than 3 million. Rods responsible for vision in dim light, cones responsible for vision in bright light. And in rods there is only one photopigment, rhodopsin is present, but in cones three types of cones cones are of three types with different photopigments. Based on the different type of photopigment present in the cones varies. Different types of cones exist based on what type of photopigment exists in that. Okay, next our topic is we are going to learn how that vision is happening and visual pigments, all those things that we can learn in next section. So with this, we can finish today's section. Thank you.